Thank you very much, Hanna, for the kind introduction, and thank you all for hosting me here. It's my first time in Mexico City, and it's really been wonderful to see a lot of friends of the Weizmann who I typically see, like Mr. Stillman here at the Weizmann now. I am here in Mexico. So what I'll do in the next 20 minutes, I want to talk to you about this big word and general topic of stem cells, and which is you probably heard about, and why it is important for treatment and so forth. So I want to present to you the field and also some parts of my own research and how the lab, and trying to show really the reality, the promise, and the problems that we have. So in the next slide, I will show you that this craving for rejuvenation. I'm helping Marcus Marcus, if you can just hit on the middle of the screen. No need for that much dark. We'll get it and work. So you think now a major challenge really we face in modern society is the fact that we live much more than we did 100 and 200 years ago, which is a great thing. But with the fact that we live longer, we face new diseases. We face increase in incidence of diseases. You can think of Alzheimer and Parkinson's that are becoming more abundant with aging. Cancer is also somewhat in a way of aging, because if we live longer, there's a higher chance of getting sort of cancer. And also, I think the demand for high quality of life is really important. We want to play tennis or climb not only when we are 30, but also when we are much older. So really the potential solution for these diseases, if we can find a source for transplantation. If we can find an easy source, we can get organs, and we don't have to look for donors and look for matching DNA. These are the usual problems we have with transplantation. That will be nice. So really the problem and the challenge, how can we find a suitable source for cells or organs for transplantation? That is very easy. So if you can see, this craving, in a way, for rejuvenation is not new still. And I really like this painting from Cranach from the 17th century, the 16th century, actually. This is called the Fountain of Youth, where you can see here the older patients on the left come to wash in the water of this fountain. And on the right side there, they come out young, healthy, and naked. So this is really a kind of a cycle of rejuvenation that we think about. But if you look, we actually, as living organisms, we have a fountain in youth of us. So if we look in development, really, we have every organism, every one of us, started from a single cell. So if you think about it, we have, our life begins when sperm meets an egg, and usually there's some alcohol around. And then we have a one-cell embryo that starts to develop. And then we have this ball of cells, the blastocyst. Then this embryo implants in the uterus, and you will see that it will start progressing to become a complete embryo that continues in pregnancy. But also, actually, already very early on, we specify these cells, which we call germ cells. Basically, the progenitors of sperm and egg are already specified very early in the development. And that way, basically, the cycle of life of these early cells goes on, and that's how we transfer the genetic material from generation to generation. And the key to understand, perhaps, this fountain of youth is understanding these early cells, because these cells have the potential to give rise to the entire embryo, including every organ in that embryo. And if we understand their secret, maybe these cells will be the source to make organs. We don't want to make an entire embryo, but we just want to make an organ. Is that possible? So scientists really were able to show about 30 years ago that if you take an early embryo, just 3 days in development, about 64 cells, and you explant it in the dish, you can grow cells which we call embryonic stem cells in the dish. These you see below, these are colonies. They are very nice, round and shiny cells, and we can basically keep them in this state forever. We give them the right hormones, they stay there. We can even freeze them and we can thaw them, and all, but still, they still have the potential. If we put them back, they will give rise to an entire organism. So they can differentiate into every cell type in our body. So this is what we call them embryonic stem cells. And I'm not talking about adult stem cells in our body. So we have, for example, adult stem cells in the skin. We have stem cells in the blood. But blood stem cells give only blood. Skin stem cells give only skin. I'm talking about embryonic stem cells. This is the first cells. You can think of this as a pyramid. This is the top of the hierarchy. And really, why, as I said, ES cells, embryonic, have become very promising, as perhaps they are the source we've been looking for to make cells, because they have this property. They self-renew, they keep their state, they keep growing and have this potential. And the next thing, we can basically make them differentiate. Next click, please. You can make them differentiate into every cell type in the body. And that is very important. So basically, these cells not only were derived in the mouse, but you can derive them from human embryos. Usually these are embryos that are donated for research. Couples who undergo fertility treatments, infertility treatments, often they have frozen embryos left. And usually about 15 to 20 years later, they can be asked, should we discard these embryos or can we extract cells from them? And you can derive human embryonic stem cells from such embryos. And again, these cells have the ability to differentiate into every cell type in the body. But if you think about what we want to do here, we're thinking of this patient, let's say a Parkinson's patient who needs these cells that make dopamine in his brain, or a diabetes patient who needs these insulin-producing beta cells. And the idea is that we want to take these embryonic stem cells. We don't want to transplant embryonic stem cells. These cells are very early on. If we inject them, they will make a mishmash of cells. They will make many cell types. We want to first differentiate them into exactly the cell type the patient needs. Meaning, if you're a diabetic patient, you will make only beta cells. If we need Parkinson's, you make dopaminergic neurons. And these cells, we want to transplant them to the patient. So that is a very nice dream, and it's important. But scientists wanted something more. The problem, no, please go back one slide still. The problem, can you go back once, please? More? Back, please, it's okay. Regreso? Yes, please. And the problem with stem cells is that if you take these stem cells that are, let's say I'm the patient, and these are stem cells donated from the embryo, the DNA is not similar. So it doesn't mean that it matches me. As you know, when you look for transplant for organs, the DNA has to be similar. Otherwise, there will be rejection. So scientists started saying it would be really nice if we can make stem cells not from a foreign embryo that is donated, but if we can make stem cells from the patient himself, that they will have exactly his DNA. And that way, we don't have any chance that these organs will be rejected. How is that possible? Obviously, we can't, if I'm the patient, I don't want to go back and to become an embryo again. That is not the goal. But this is possible in a different way. If we think, what is the difference in our cells, in our body? Next slide, please. And if you think in the cells in my body, I have the same DNA in all my cells. My heart cell and my liver cells are different, but they have the same DNA, okay? So I have one genome. The difference between the different cell types is how the DNA is organized. So if you look at the next one, we have one genome, but we have something called many epigenomes. What does it mean? The DNA, every cell in my body has 6 meters long of DNA, which has to go inside 2 microns, okay? And the way this is done, the DNA that is very, very long, it is wrapped very tightly. And there's a regulation. You can think of this DNA. You have neighborhoods that are very, very tight, so the genes there do not get expressed. And you will have neighborhoods that are loose, are open and happy, and there the genes are expressed. So the difference between my liver and my brain is about which neighborhoods are open and which neighborhoods are tight. So I have again, and this means epigenetics. It is how the DNA is wrapped. It's above genetics. And so we have one genome, and we have many epigenomes in our body. Basically, every cell type is a landscape of epigenome. So really the next question was, can we then take a cell that is differentiated, like a skin cell or a blood cells, and basically open the DNA completely? Everything, or like you can think of a computer, hit the reset button and go back to become a stem cells. Can we go back? And a very important experiment showed that this is possible, and that was the success in cloning Dolly, or even before that, the frogs. Why this is important? Dolly is important. It's not about making another more sheep or whatever, but it showed what you do in cloning. This is an egg from the donor. You go in with a needle. You take out the DNA. You take a skin cell. You take the DNA of a skin cell. You put it into this emptied egg. You put it in the uterus, and you get an entire organism. So this experiment really showed that the egg has somehow ability to completely erase everything and restart and reset development every time. That was the importance of the cloning of donor. So this experiment was very important because it created this scenario where basically if we're talking about a patient who needs stem cells, maybe we can make stem cells from the patient himself. And the way to do it is basically we thought of maybe we'll take a biopsy from this patient, take skin cells, go to a female donor, ask her to donate eggs. We'll take out her DNA. We'll put the patient's DNA in. We do not and cannot, of course, implant this. We don't want to make a human being inside the uterus. We just grow them in the dish for 3 days and derive stem cells from this blastocyst. And in this case, the stem cells are completely made from the patient. So that was really great and showed that there is a way to make stem cells from the patient himself. But in science, as you know, many times you solve one thing, but it opens many other problems. There's a lot of problems with this technique. First of all, to get human eggs is invasive. It's hard. You need women to get through hormone therapy, which is problematic and has side effects. Also, for example, the Catholic Church mainly has an ethical problem with these experiments because life, for example, in Christianity is considered that it begins already after fertilization. And are we really creating some kind of life, destroying it to save another? It's a legitimate argument. It's a problem. By the way, in Judaism and Islam, they don't have an ethical problem because life begins with the heartbeat, which is much later than these stages. But I would say as a scientist, my problem with this technique, it is technically obsolete. It is extremely inefficient. you need about 100 to 200 eggs to make one cell line. So if you think of economically, even in the future, this is not a very valid way. So then this raised the question, okay, instead of using eggs, this problematic part, maybe we can understand what are the ingredients in the egg that do this activity. And maybe we can find, 1, 2, 3, 4, 5 genes that are expressed here. And if we basically take them out, put them on the skin cells, maybe we can convert them directly into a stem cell in the dish without using any embryos. Is this possible? And really there was a big race, and 10 years ago, the group that really succeeded in this was the group of Yamanaka in Japan. He discovered 4 proteins, OCT4, SOX2, KLF4, CMYC, the name do not matter. These are proteins usually expressed in the egg. And he showed if you impose them, we just express them, we have ways to express them for about 2 weeks in the skin cells, the cells will convert in the dish and become what we call iPS cells, stem cells. And the iPS cell stands for induced pluripotent stem cells. Induced because we are inducing them. So I talked about ES cells, these are embryonic stem cells from the embryo. iPS cells are induced. Can you go back? Sorry, just one. And this is what you start, this is what it looks. You can start with a skin cell. These are the fibroblasts. You can see they are very, very flat. And the end product you get, again, these are mouse iPS cells. You can see these are the cells that are shiny and growing into colonies. Every colony has about maybe 100 cells of this. So really, this was an amazing discovery. And to show you that these cells are really functionally stem cells, one thing we can do in the lab, and if you hopefully visit the Weizmann Institute and visit my lab as well, you will show you have these microscopes and you have joysticks on the side, and we can work with micromanipulation. So we can take, for example, stem cells in a needle, for example, stem cells from a mouse that is albino, or we can take stem cells and make them green fluorescent. And these are, you will see it here, sorry. 1, 2, 3, and we go with the needle inside another embryo, and we can inject them into the embryo and implant the embryo in the mouse uterus. And what you will see next, and that when we transplant this embryo, next slide please, you will see that if we transplant green cells into the mouse, you will get green mouse completely. It's control. So this really shows you, we start with a skin cell, and we can completely go back and make stem cells that can make an entire animal, showing you the power of this technology. So this is very important, iPS cells. And next slide, please. This is just to summarize the cycle that I showed you. We're starting with a skin cell, going to a stem cell. We can inject it and make an entire animal out of the thing. So what is this important for? What can we do with this technique? So one thing we started to do was showing we don't want to make an entire embryo, we want to make cells for therapy. So one thing we did, in the next slide please, this is to show you what this looks like. Okay, hit please the start button. You're now going to see a movie of the cells. Just hit in the middle, please. Yes. These are the skin cells growing for 2 weeks. Growing, and suddenly you see this colony here. Okay? We're going to play it again. Please hit again the space button. One more, please. And you will start seeing skin cells proliferating, and a very rare cell will appear there. And these are the iPS cells that we pick to make stem cell line. And I always like showing this video, and I work in this field 10 years, and I always say, this is amazing, but it's not a miracle, okay? So this is a biochemical reaction that we would like to understand, and that's one of the things that my lab does, where we want to really understand what are the enzymes converting the cells in their identity. And we ask, how, why, and what. How does this process work? How do you change the DNA from a skin cell to become a stem cell? Why does it work? How similar it is to what's happening in the embryo? And these are very basic biological questions we like to ask. First, because we call ourselves geeks or nerds. We want to understand biology. And also, we believe that's the way we can understand if the cells are normal and whether they can be used. And then I want to show you one discovery that we did in the lab. No, please go back. It's good. Yes, perfect. Reprogramming process is very inefficient. And about 1% of the cells, no, please go back. Sorry. And we're going to watch the video. And if you can just hit on the screen of this one. No, it doesn't work. Okay, that's fine. If you can, let's try one more time. And wait. It's okay. So we want to take skin cells in this program, and usually about less than 1% of the cells succeed in going back. Okay? You saw in the video, it's very, very inefficient. And we were asking, why do most of the cells fail? And can we make the process not random? Meaning, every time we do the cell with these 4 factors, we want them to go back to become to an embryonic state. We want to be completely non-random. And what is preventing it? And what we discovered, we discovered that cells have basically behaved like a car. The cells have the gas pedals. These are the 4 proteins we put. But they also have handbrakes. They have a protein called MBD3, it doesn't matter, that prevents them from changing and going back. And if we release the handbrakes, it's like a car. The car drives very smoothly. So we can basically get 100% efficiency in 8 days. It becomes very, very efficient. And this what let's see if the video works. Now, yes, it is working. Just leave it. And you can see that, no, please go back. It will start. It's starting. And if you look at the skin cells, it started before, but can you hit just once, please? Doesn't matter. So you're going to believe me that we can make the process very, very efficient. So we have that we can move from 1% efficiency to 100% efficiency, okay? And this is important for a lot of reasons, also for technically, but also for studying the process. No, please go to the Hanukkah. Okay, next slide. Okay, yes, please. So what I'm telling you is you can see here in 8 days, we can have all the cells becoming iPS cells in 100%. And this is really exciting. Why? Because what we are also trying to understand, what happens to the cells during these 8 days, okay? And we call this indeed Hanukkah projects. We have 8 days. In 8 days, we have 100% efficiency. And every day, we harvest the DNA from these cells and look how the enzymes are working. And we can really, with this mapping, we can understand what is going on these cells as they are changing and really understand the fundamental biology of this process. So the next thing we were trying to do is to show really can we use these cells for therapy. And what we did, we took a mouse model that have a mutation for sickle cell anemia. This is a blood disease, a genetic disease, that there's a mutation and the red blood cells break. And that causes a lot of damage. It's still very common. About 10% of African Americans actually carry this mutation. So what we did in this mouse, we took the skin cells, then we infected them with these 4 factors that convert them to become stem cells. And we made iPS cells, these are, that are genetically identical to the mouse. The next thing we can do, the nice thing about stem cells, because the DNA is very open, very easy for us to correct a mutation. So what we did, we genetically engineered new DNA, and we can remove the DNA mutation and put instead of it the same sequence that is corrected. So now we have basically genetically corrected self-IPS cells. Because we don't want to give the patient, the cells that are diseased. And then in the dish, we made blood cells, bone marrow. We transplanted them into this mouse, and we were able to show that 80% of the mice had basically corrected of this phenotype in between them. So really showed that this cycle of treatment is possible to do with these IPS cells. Again, showing the power of this technology. And now, and the major goal in the field is, of course, to make many cell types for transplantation. And as you can think, as I told you, we want to inject only mature differentiated cell. And there are hundreds of labs in the world that specialize in making different cell types. There are labs that specialize in making skin cells. My lab actually works on making human female eggs for infertility problems. Labs work on making insulin-producing cells. And they basically, our labs and other, define protocols of how to move forward step by step until we reach normal, mature cells and those cells that we want to transplant. And this field is very young. It has been 10 years now. But we are very happy that, for example, the first clinical trials in Parkinson's are starting next year in April 2018. And I think we're going to hopefully see a lot of success and new trials with this technology. So what I showed you in the first part of this talk, that we can make stem cells from the patient himself, from the adult, from skin cells. You can make it from any age, no problem. And you can make cells that are important for transplantation. But another really important thing, and not only for transplantation, is that we can use these cells to study human diseases. How is that? Let's think today if we want to study how does Parkinson develop or how does diabetes develop. We can use mice, and mice are very useful, but they're not human beings. If we want to take human samples, it's very hard because it's invasive. You want to go inside and get a biopsy from the pancreas. You're going to get very little material. And actually, the situation is much worse because by the time the patient is diagnosed with the disease, the cells are already gone. They're already dead. So if we want to understand why this disease and why is this deterioration starting, it's too late. But what we can do with technology for a lot of diseases, genetic diseases, where you can think of Tay-Sachs, cystic fibrosis, even in Alzheimer's and Parkinson's, there are forms that have mutations in families that they get Alzheimer's at the age of 20 or 30 even, really bad cases. And we can go to this patient and get just one drop of blood and make from the patient stem cells. And these stem cells will have exactly the mutation and the genetic identity of that patient. So if you think about it, we have, let's say, a normal person, we have the ALS patient, we can make these IPS cells from the patient himself. We can grow them unlimited numbers, we can freeze them, we can make them glow red when they die, we can make them glow green when they secrete insulin, we can correct a mutation, we can make 2 mutations, we can compare to the healthy brother, a lot of flexibility with human materials. And then, let's say we're studying Alzheimer's, we can differentiate these cells into neurons and start asking what genes are being changed during this death of the cells. What is happening to the cells and why are they so stressed? So it really shows you that this is a very relevant way to look at human diseases in a dish, where we're usually very, very limited because we don't have enough samples. With this technology, we can study human diseases in a much better way. Pharmaceutical companies are interested very in this because they have chemicals from plants or drugs they synthesize, really millions of them. And of course, you cannot just randomly screen them in human beings because it's dangerous, it's extremely expensive. But the fact that we can grow, for example, neurons or beta cells with large quantities, they can, with robots, screen these chemicals and test, can we find now drugs from plants that increase insulin secretion or drugs that improve Parkinson function, and many, many different examples. So really, the stem cells are becoming, these human iPS cells are becoming a platform for drug discovery as well. So just to summarize my talk really, and just to show you, when we talk about embryonic stem cells and iPS cells, often, yes, a major goal is tissue transplantation and regeneration, but they have also many other applications. Their application to look at reproductivity and fertility problems. As I showed you, they can be useful to make human disease model in the dish outside the human body, and we can use them for drug toxicity and screening to find new drugs. Now, I showed you a lot of the reality and the promise of this technology, but I also want to finish my talk about saying that also we should be careful. And we should be careful because there's a little bit in the media sometimes, the newspaper sometimes, stem cell hype, and a lot of making promises or unsubstantiated treatments about stem cell treatments that are actually, if they don't work and they just, take your money, that's fine. But if it's dangerous, we should be careful. And I'll show you this example. This is about Liz Taylor about 10 years ago. You can see these newspaper, how that she's looking very, very well because she got some kind of stem cell treatment. If you Google stem cell treatments, you will find, for example, this company that tells you we inject you with Swiss apple stem cells. I have no idea what the Swiss apple stem cell is. And actually, it tells you if they inject you, you would look as young as Bill Clinton. I don't know why they chose this, yes, but that's their website, you can check it. And again, of course, the idea is I'm not dismissing any of these companies, but I just want to create awareness that please be careful and consult with your doctors and people that, yes, there's great promise and great progress, but you just be careful not to do things that are not proven or too early. It can be dangerous. And that's just my slide. My group, I've been back at the Weizmann Institute now for almost 7 years. It's been a really, really pleasure. We were fortunate to meet Ilana and Pascal Mantou, who were kind enough to donate money to start my lab and built it. Since then, we've established a team. We're about 14 members and able to get grants and funding from different agencies to be able to do the research that I showed you. And the final slide, really, I just want to thank you for being here tonight, for showing your support and interest in science in general, in the Weizmann Institute. And if you're really ever at the Weizmann, your friends or family, please visit us. I'm talking about us, my lab, visit me. If you have kids, grandkids, please bring them because we want to motivate also the next generation of scientists so that they understand that the lab is a nice place, it's not boring. And with that, I finish and really thank you very much.
Preguntas? Shimon, English.
English, yes. Considering that you can cure so many diseases, as this technology will improve, what do you think life expectancy can be?
So, no, I wish that is true. I would like to say that, for sure, at least life quality will improve, which is very, very important, to just have a very good quality. And I'm sure, life expectancy, with these, in 30, 40 days when these treatments become routine, it will increase in some level, but, it's not going to change very dramatically. We cannot replace an entire brain. We cannot replace entire blood system, and I do not think so. Not yet, but unless never. But as I said, if you can really improve the quality of patients, and of course you can hopefully save lives of cancer patients as well, because then if you think about many patients now, if you give them chemotherapy, we're not giving chemotherapy strong enough because we are worried that we will ruin some organs. But if you know that, I have backup, I can take his skin cells and I'll give him the liver again, I can then hit harder with chemotherapy. So this just shows you kind of indirect scenarios where this technology will have implications. And time will tell. I don't think there are going to be a lot of failures as well. But I think I very much believe in this technology, not only because I work on it, but I think really to take a cell and you make an entire mouse out of it, it shows you that the potential is there. We just need to control it and channel it in the right way. And we will see how it goes.
Thank you, Doctor, for your brilliant presentation. I have a question. In the early future, gonna be possible to a mutation, a cell with a mutation like BRCA or P53 sent to apoptosis, and after that replaced with a new cell without the mutation, gonna be possible?
So I think, yeah, so the question is about can we correct a certain mutation in a certain disease, and it's definitely possible. I showed you one example in this case of the sickle cell anemia to correct the mutation. And this is now even becoming much easier. Some of you might heard there's a new technology that it's something called CRISPR, Cas9. It's basically there are very efficient enzymes that you can really direct them to cut exactly in certain loci where you want in the genome. So really it's much, much easier these days to, even in human cells, to correct a mutation or delete a gene or insert. So the stem cell technologies and the genetic engineering, actually technologies have always gone side by side and relied on each other. And we expect to see more and more application of this.
There is the serious problem that is for many people. How do you, is there a way to help them?
So I was asked about the celiac problem, which is a problem in that basically it's some kind of a very strong allergy in the intestinal epithelium to gluten, usually. And this is kind of dangerous because if it goes over time, it can develop to cancer. So it's a very specific cause. I would say there is a bit of there are big questions in terms of we don't know really why this disease is developing. And but at least if from those patients, we can take skin cells, not my lab, other groups take skin cells, make stem cells, and make colons, like intestines, in the dish. And they are starting to study this reaction, by exposing them and how they're responding. So I think it's definitely going to be a learning from being able to study this disease, because mice actually don't have gluten. There is no celiac disease in mice, and this shows you that the power that we can now have human intestines in the dish from those patients, and they behave like the disease. And I expect we will learn much more about this disease from these models. Yes, please.
Gracias. I have two questions, very related. Can this be applied to spinal cord and to brain cells?
So the answer is yes. So these cells can make every cell type in the body, including different types of brain cells. for example, in the case of Parkinson's disease, where it's a disease where people get very strong tremor, what happens there, it's a very specific region, very small in the brainstem. There the neurons die, and we know exactly where these cells are. And there, the first clinical trial that is starting is basically making these, they're called dopaminergic neurons, and transplanting them under the MRI exactly to that locus in that region. So that is already happening. We cannot, although we can make many types of neurons, we cannot replace an entire brain of a human being. In many cases, like Trump, for example, it would be helpful, but it's not possible, unfortunately.
Today we're thankful to him.
Well, yeah, but on other days. So we can, and that's a limitation, right? Because the brain is connected to other places. So the spinal cord, again, it depends on what the type of the injuries in the spinal cord. So there are some injuries that, you have complete destruction. I don't think it's going to be possible to do it. But in some cases where only specific what's called motor neurons there, and there's some diseases called SMA, it doesn't matter. There are, I think it will be possible, and there are already groups working on it. So as I said, it's and maybe, yes, maybe as he or she correctly said, maybe in the future you can replace an entire brain, maybe. But
thank you. I have two questions.
yes.
And the first one is, are there differences between the iPSC-derived mice and the wild-type mice?
Uh-huh. So let me answer your first one. So yes, so there's this big questions really. We make these iPS cells, and how normal are they to embryonic stem cells? And if we make these mice from them, are they normal? And the question, yes, they are normal. There's now been very long follow-up on these mice that we make them. They are normal. They don't get cancer. They're fine.
Okay, and thank you. And the second one is, I think that this field about transplantation is one of the biggest challenge, because you don't know if once you put the cells derived from iPS cells, they will respond or answer to this like in the same manner of the wild type cells. Like if they can synchronize their biological clock with the biological clock of the
host tissue. That's very good. So again, if we want to transplant, yes, we don't transplant iPS cells. We transplant their derivatives. That's definitely. But we already know that many times after you get transplant cells, they do synchronize. For example, if you inject, you can take liver cells from a donor, okay, and inject them into my liver, and that's how we do transplant, and they will survive. They will synchronize. In beta cells, actually, they have clinical trials where they took they took insulin cells from about 10 patients who died, harvested these cells, and transplanted them in the pancreas, and they lived. They were synchronized. As I mentioned, however, the problem, this is not feasible as a treatment. So, but in the heart, for example, that's what you're saying. So you have the heart is beating, and it has if you integrate these cells, they're sensitive. Are they going to get the same electricity or something? So it remains to be seen. The nice thing, as I said, like about every cell type, there are tens of labs that are specializing. How do we make the heart cell? How do we deliver it? If you think, in the case of the liver, it's easy. You just take a mesh of cells, you can transplant them like this. In the heart, you need perhaps to make like a patch, and you put it on the tissue. And so, for example, many labs collaborate with engineers who can use 3D printers, and then on these things you grow the heart cells. So you'll have a mix of kind of material, some kind of plastic with the living cells, and transplant them. And so just giving you examples, so the field is really expanding because of these many different questions, and a lot of engineering is involved as well.
Hello. My question is that if we're talking about like monogenic diseases, like sickle cell anemia, it's like kind of easy because we can just edit those genes using a CRISPR-Cas, something like that.
Yeah.
But when I saw like the Alzheimer, it was like really, I don't know, I would like to know how the strategy, because we're talking about a disease with plenty of factors that are affecting that. So there are important genes there, but they are not deterministic.
Yeah.
So I don't know how is the strategy that you are taking to.
Yes. No, that's very question. So to kind of reword your question, if you talk about a certain disease, you want to correct it. Some diseases are, let's say you have the mutation, like sickle cell anemia, you're going to be sick immediately after you are born. Or cystic fibrosis, you have a mutation, it's clear. Versus if you look at Parkinson's disease, most of the cases it's not mutations. And not only that, it takes a lot of time to get Parkinson's. It's not like cystic fibrosis that you get immediately when the cells are made. And so just giving you an example, how the cell fields expand. So if you go to although there are rare forms of Alzheimer's, there are some cases that have 1 or 2 mutations, and you will get the disease in about 20 years, which is extremely fast. But still, it's 20 years. You're not going to wait in the incubators 20 years. But what scientists do is find out what happens. For example, if you stress the cells, they will start showing signs of Alzheimer's. So basically, there you have this new field where how do we get the cells to behave like diseased ones much quicker than they usually do? So there are quite like tricks how to do it. Also, for example, it's like do you see the movie? What's called something Benjamin Button? Okay. So there's a disease. Even though it's exaggerated, there's a disease, for example, called progeria, where aging becomes much faster. So you look at them they're like 14 years old, and they look like 70 or 80. So basically, you can take Alzheimer mutation, mix it with the Benjamin Button mutation, and you will get the disease much faster. So it just shows you scientists are trying to think of tricks. It's models. It's not, and it's humans. We are limited. We can't do what we can do with mice. But we're trying our best with the genetic engineering to see, and it's a model. It's not perfect. I don't think every disease is going to be modeled, but many do. And
talking about these like multifactorial diseases, are you taking like any approach like metabolomics or something like that to look how's the metabolism there?
Yeah. I mean, those scientists, my lab does not work on this, but my colleagues in the field, for example, those who are working on liver metabolism, cholesterol, or fat, they apply. Then once you have these cells, you can do whatever analysis you want. You can look at gene expression, you can look at metabolic profiles, you can look at under the microscope. You can do whatever you want with these cells, and that's the big advantage. And if you need more cells, you don't have to go to the patient. You thaw them from your freezer and make more cells.
Yes, hi. For patients with kidney failure, is there any hope with the new stem cell
Of course.
development?
It's a big part in the field of trying to make kidney cells and kidney organs. And the kidney is a tough organ because it's actually, compared to the liver, it's much more complex than the liver. But there's a lot of progress in the last 2 years, really, of being able to make the early embryonic progenitors, that's what's called the buds of the kidney, that's usually during the embryo. And these buds can go to entire kidneys. It's still far from being perfect, but I think that the breakthrough has been made in getting these buds. And I think in the next 2-3 years, we're going to see more. I think still it's going to be much harder than liver cells and I said Parkinson's cells, because there in the kidney we probably need the entire organ. But the potential is there, and we'll see. There are those, including our lab, that actually try to take these buds and then transplant them into pigs. So you do part of it in addition, part of it. Kosher problem, we'll leave that aside. But if we can get a kidney, I think that could be another thing. And just shows you that this field is trying to solve a problem by many different ways. And we don't know which one works, but it's nice. We try many things, many different labs, and we see what works.
Can you create a new kidney from the stem cells?
Yes, you can. That's the whole point. That's what we're trying to do. And so in the mice, you can get an entire kidney. But as I said, we want to get either an entire kidney. I cannot make an entire human being full, but I want to make it either in a dish or just make some part in a dish and then go to a pig and get an entire kidney. That's what we're trying.
When you see that the microenvironment is the problem in an example like diabetes mellitus type 1, you also are transplanting the cells that are dying and the lymphocytes? Because the main problem there are the lymphocytes, not the beta cells.
So if you talk about diabetes, there are two types of diabetes. There's type 2, which is the more common type, where usually in older patients that we have just diabetes, slowly the beta cells start less secrete them. And there the solution is relatively easier because you can make these beta cells, new beta cells, and transplant them. And you mentioned there's type 1 diabetes, which is diabetes that happens much earlier, and there the immune system attacks the insulin-producing cells and kills them. And there, as you said, if the treatment is going to be more complex because not only you need to give beta cells to these patients, you also need to prevent the immune system from killing them again. Okay? And again, there are different approaches that are now being very close to the clinic. One approach is a company called ViaCyte, which is very clever. And unlike in the case of insulin cells, they don't have to be in the pancreas. If I actually put the insulin cells, I don't know, in my armpit or here, they will be fine because they just need to sense the glucose and secrete insulins. And what they are doing, they are putting these cells inside some kind of a capsule that cells cannot go inside, but they can secrete from the inside toward the outside. So that way the immune cells cannot kill them. But I would say one advantage, again, of this disease, let's say worst case scenario in type 1 diabetes, let's say the patient gets a capsule, he manages to attack it after 5 or 7 years. Again, you will thaw his cells and give him another capsule every 7 years. Worst case. But I think it will be better. And again, we don't know. We'll see. I'll just ask here the question, and Mr. Yes.
Thank you. For example, in the case of Alzheimer's disease, in which there's been a chronic environmental, the whole thing has neurofibrillary tangles and amyloid angiopathy and whatever. But what's the chance of a normal cell that's, or a stem cell that has all that potential being introduced into a tissue that's been chronically damaged and it's locally, it has many epigenetic changes. Or chronic inflammation or whatever.
No, it's definitely. So you're talking about in the cases of in the case of Alzheimer's, it's probably it's there. The cause, I think, of treating it with cell therapy is I don't think it's feasible, because as you said, it's too broad. It's many types. There, I think more the interest in the field is making iPS cells to study why the disease is happening. And I think there is actually to find drugs to prevent the deterioration. But if you take a different tissue, actually in the case what I showed you in sickle cell anemia, Okay? So the bone marrow itself is very, very diseased. When you take the cells that you corrected, they actually take over. Because they are much healthier, they survive. And the diseased ones go away. So actually there, you don't need to give irradiation to the bone marrow to clean it, then transplant. You can just transplant the corrected cells, and they survive because they have selective advantage. If you think, again, just showing you every disease has a scenario. If you think of leukemia in cases of cancer, there you have, because the cancer cells are proliferating, the healthy cells do not have selective advantage. So you actually have to give chemotherapy first, irradiate very strong, and then transplant. So again, just depends on the disease and different scenarios that happen. Louise, do you have questions?
You have a patient who has a disease. So you take a cell from him and modify it and put it back. The disease has an origin, some disease which is in every cell. When you put it back, how do you neutralize the disease which he had?
Yes. So that's one really important. This is a very valid point, and that's why the gene correction we correct the DNA. So if it's a genetic disease, we have to correct the DNA, and we can do it. We can just basically what we do. When we have the stem cells in the dish, we synthesize the DNA that is healthy. We give them an electric pulse. The DNA goes inside, and it flips with the other one inside and takes it out. Of course, we have to check those. We can check those that were corrected, and those we will use for therapy. So that's why it's very, very, gene therapy is heavily involved in what we're doing. There's gene therapy, that's why we call it gene and cell therapy. We have to do both. Yes. So,
I have a question about the immune system that attacks different organs, like thyroid or the cells of your skin that become vitiligo or something like that. What do you do with it?
So you're asking about really a lot of types where the autoimmune, where the cells of the immune system attack our organs. And actually, what you asked earlier, the celiac disease is kind of an autoimmune because the immune systems are overworking. I would say a little bit for what we do here, it's less relevant actually, because in the case of the autoimmune system, I would say we have to first understand why the immune system is attacking the cells and replacing them. Because in these diseases, to replace the bone marrow completely is very aggressive. That's why we less. So that part is less related to this type of research.
So, well, let's say that you have a genetic disease that is generalized. Let's say you have a genetic disease that it's generalized because, well, from what I understood, this treatment only works on very focalized genetic diseases, such as Alzheimer or even like, well, I don't know, Parkinson.
Yeah.
Do you believe that it could someday work on very generalized genetic diseases such as, I don't know, Down syndrome, even if it's detected early on the womb or something?
Yeah. So we're asking. It's a great question. We're asking about where mutations like Down syndrome, where basically you have an extra chromosome in all our cells, and what happens with it. Of course, we can. And you're talking about scenario where we didn't detect. So the child is born already. Okay, we have the child. And yes, I think it is relevant because, yes, we cannot replace every cell type in that patient, but at least, for example, we can alleviate some of the cases. For example, Down syndrome, they have problem in blood as well. It's not only mental retardation, and we can take cells from them and remove the extra chromosome and give them a corrected bone marrow. So basically, we can find ways to alleviate the disease. There's another genetic disease which is called Fragile X syndrome, which is also common in males. It's an X chromosome diseases, and it comes a lot with mental retardation. And there are ways to kind of engineer the neural stem cells that are key in this point to express again this protein. It will not rescue all the phenotypes, but at least in some of the key phenotypes that they have them. Okay, thank you.
Thank you. Don't go. Stay here.